So first, I'm just going to give an intro of the seminar series, and then we'll intro for Emily, and then we'll get started with the talk. So my name's Robert Driver. I'm a postdoc at Duke University, and I want to welcome everyone to the AOS Student Research Grant seminar series here. And before we get to our speaker today, I will introduce the seminar series as well as the AOS grant, which inspired this series. And so each year, the AOS offers funding to students and early career researchers with the AOS Student Research Grants. And each year, the AOS Student Research Grant Committee receives nearly 200 applications for this, making it highly competitive, even with when compared to other society-based small research grants. So I want to acknowledge the substantial amount of effort that goes into writing a successful proposal. Winners are able to describe a project that advances the field of ornithology while still remaining feasible as part of often as part of a graduate dissertation. So the AOS Student Research Grant Seminar Series, which we are all here at today, celebrates the challenging and innovative work of the AOS Student Research Grant recipients by sharing AOS funded or related work to you guys, the AOS members. So each week we are showcasing the research or research plans of a 2023 or 2022 AOS Student Research Grant winner in a seminar style format. We are in week 10 now, I believe, 2/3 of the way through. We'll have one seminar each week for 15 weeks until May 5th, so coming up on it. So I want to thank everyone for being here, thank the AOS and AOS Student Affairs Committee for support and input, and thank Chris and Handel and Ramona Fletcher for all their support. And before we turn our attention to Emily's research, I want to mention to put your microphones on mute, although I think that's the default setting. If you have any questions, there will be time for a Q&A session at the end of Emily's talk, and you can participate by either raising your hand icon, and then I can unmute you, and you can speak or turn your camera on if you would like, or you can type the question either in the Q&A chat box feature, which is at the bottom of the Zoom screen, or you can also use the regular chat box. I'll check all these different ways to ask a question at the end, so feel free to use whatever you're most comfortable with. And then, sorry, yeah, Matt, did you mean to raise your hand? I can have you ask a quick question if you do.
Hi, Matt.
Uh, but looks like Matt's not unmuted. And Maria, raise your hand. Maybe these are accidental, so I'll just move on. If there's a question, you can feel free to message me in the chat, and I can try to address it. Um, oh, right. So right. So Jason, if you raise your hand, I can unmute you and have your camera turn on. So so if you just raise your hand, I can enable those things for you, and we can ask a question that way. It just requires a little extra step. Thanks for clarifying. I should put that in my intro. And I also like to remind everyone that this is an AOS sponsored event and that all participants are required to follow the AOS Virtual Meeting Event Code of Professional Conduct. And I will post a link to the AOS Virtual Meeting Event Code of Professional Conduct in the chat. And also please be aware that we are recording this talk. And one more link, which will be in the chat in a moment here, is a post-seminar evaluation form. It would be really great if you could take a moment to fill that out. We're trying to make this seminar series better and better each week. So to fill that out is very valuable for that, and I'll put that link in the chat. Okay, so now, for our seminar today, Emily Griffith is currently a Ph.D. candidate at the Drexel, Drexel University Academy of Natural Sciences of Philadelphia. So Emily's academic career began with a dual major bachelor's degree in zoology and psychology from the University of Florida. And then Emily went on to receive her master's degree in biological sciences from Northern Michigan University, working with Dr. Alec Lindsay. And here, Emily researched size and plumage color in long-eared owls. And her research is published in the Journal of Raptor Research, as well as an upcoming publication this summer in the Wilson Journal of Ornithology.
It actually just came out
Oh,
online
just
yesterday.
came out yesterday. Okay, never
So
mind.
good
Not
access.
upcoming.
I know. I was like, I should have emailed you.
yeah, no. So as soon as this seminar ends, everybody head over to the recent Wilson Journal publication. So Emily gained valuable museum experience during her master's degree as an assistant curator of ornithology at the Northern Museum of Zoology. And Emily continued with her museum work experience and joined the lab of Dr. Jason Weckstein in the at Drexel University in the Ecology, Evolution, and Earth Systems program at the Academy of Natural Sciences. And in addition to researching and preparing museum specimens, Emily has worked extensively on duck genomics, releasing multiple duck assemblies in a recent publication in Biodiversity Genomics. And in addition to the AOS Student Research Grant that Emily won, which we all know about because that's why we're here, Emily also won additional society-based grants, including the Early Systematist Award from SSB, the Chapman Grant from American Museum of Natural History, and the most prestigious grant offered by the Wilson Ornithological Society, the Luis Agassiz Fuertes Grant. Okay, I'm excited today to learn about duck plumage, and so I'll now turn it over to Emily for her talk entitled Investigating the Evolutionary and Genetic Underpinnings of Dichromatic Plumage in Waterfowl.
Thank you very much, Rob. I know that there's a lot of handful of words there with the Academy of Natural Sciences and Drexel.
I mess it up every time, so I'm sorry.
It's a-okay. We mess it up too, if that makes you feel any better. So I'm super excited to talk with you guys today just about ducks and dichromatism and genes and all this other fun stuff. And Rob has already done the hard work of explaining what my background is. So I started out in museums probably about 10 years ago, where I was working on tarsal spur evolution with Rebecca Kimball. I'm at the University of Florida. Then moved on to Northern Michigan University, where I worked with long-eared owls, mostly migrating through Whitefish Point. And now I'm here at the Academy of Natural Sciences. And with all these photos, you might see white cabinets in the background. I promise there are white cabinets in the background just to the left of me on there. So museums are really my home for this kind of research. And my research, if you have heard my name before in the AOS context, it might be because I've designed the Fun Run shirts for the last 3 years. So we'll see if this continues next year. But if you have participated in one of the fun walker runs the past 3 years, you might be having a little cute bird on a shirt. That was me. So moving on to my research covers kind of a wide variety of topics, ranging from sexual selection, like I was saying with those tarsal spurs, plumage color, which Rob mentioned, the fluorescent plumage color in owls is that paper that just came out a couple of days ago. I also work on hybridization. I'm currently working on a hybrid project with orioles, and then systematics. And systematics is really this kind of framework that we view how these traits have changed through time and looking at the relationships of these species with one another and their similarities and differences. I'm going to be talking about all of these topics today in the frame of sexual dimorphism. So this basically concept that you can have differences between males and females of the same species in either size or appearance. So these stag beetles are a really good example of size dimorphism, kind of back in the early days when we had naturalists. They might have thought that these are two completely different species, but they're not. They are the same species. That's just the male on the left and the female on the right. What I study, especially in ducks, is sexual dichromatism. So that has to do less with size and appearance, and more just with the plumage in general. So that is a male mandarin duck on the left, with all that kind of like shiny green on the head and that orange and the purple on the breast, and those like orange tail feathers in the back. And that's a female mandarin duck on the right. She, as you can see, is a little more gray, blends into the background. She looks more like the rock that she's standing on than the male does. And the theme of today's talk is all centered around duck dichromatism. And thankfully, I'm very fortunate that I don't have to explain to any of you what a duck is. That's kind of the beauty of this research. But what you might not know is that those green feathers on the top of the head there are actually caused by melanin. So these pigments that we attribute to like the color in our hair or our skin, these are also driven by melanin. And the reason why this melanin on the head of the mallard looks green and not black is essentially these little cells will organize to the end of a feather, and that allows light to refract on it in different ways. There's actually a really good talk earlier in the series about the science that goes on behind this iridescent plumage. But for those of you that did not have the opportunity to see that talk, a very quick SparkNotes version is that you have essentially these melanin-filled pockets in a feather barbule. That's that bottom row of feathers on the left. And when they're just kind of free floating around, you have that typical dark black plumage. But eventually you can have so many of these little melanin filled pockets that they'll organize to the outside of a feather. And that allows light to refract off of it in different ways, which can give a blue or green or purple appearance. And so that's why we can attribute this very complex phenotype to a very simple mechanism. And on a broader scale, we're really looking at the evolution of these secondary sexual characteristics. And when we think about the evolution of these male-like traits, we'll say, we think about males gaining them through time through sexual selection. So females seeing a trait and they're like, oh, okay, I like that. And then, wow, now the males have it. For our example here in birds, peacocks are kind of this beautiful monstrosity we have where these traits can kind of get very, very out of hand. And we might call that Fisherian runaway selection, where the dimorphism is just so pronounced between the sexes, assumedly as a result of sexual selection and female choice. And so when we think about ducks and we think about why like a mallard might have a green head, this very bright and flashy characteristic, we'd think about maybe like a proto-duck, a duck common ancestor, having this kind of dull and dark plumage that blends into the background a lot. And then males will then gain their bright plumage on the top of their heads through female choice. This would be a hypothesis about sexual selection. But we actually have evidence when we look at these birds in the wild that contradicts this hypothesis. So I showed mandarin ducks in one of those first slides. This is a female mandarin duck that is developing male-like plumage. So she is starting to have those orange feathers on the back of her tail there. And we also have on the right here another example of a mallard where it's this female that's starting to have the black butt that males have. It's starting to have a little bit of green. And you have just these birds that really fall outside of this binary of male or female. I'll also say today is March 31st, which is International Trans Day of Visibility. And so if you want a fun fact to share with your friends today, there are ducks that do fall outside of the strict binary of having both male and female-like characters. So cool fun fact for the day. And we do actually know why this happens. So in a series of experiments that happened in like, I think the 50s or 60s, that would probably never pass an IACUC review board today, they took out essentially the female's source of estrogen, their ovaries, and found that if you remove that source, they would molt into male-like plumage. And we have these examples in nature of birds that are mallards, for example, that have this male-like plumage, but don't have testes, and they don't have ovaries either. And it's this absence of estrogen that allows these birds essentially to have this bright plumage. And so on an evolutionary perspective, we ask if females have always had the capacity to produce this colorful plumage. Is it not just a male-like trait that's been gained through sexual selection? And the story becomes even a little more complicated when we look at hybrids. If you learn one thing from this talk and one thing only, it's that ducks hybridize a lot. And it makes my job in studying their evolutionary history a little more complex, but also very fun. So that is on the very top left, a mallard and black duck hybrid. So mallards and black ducks are both very closely related species. And you can see that this hybrid kind of has like a little bit of green on the head, a little bit of black on the butt, but is kind of, again, in this intermediate state between what we'd see as a monochromatic dull species, so that American black duck on the right, and a dichromatic species, which is your mallard with the green head in the males and the females being this brown drab color. There's another really interesting hybrid that also lends itself to this story, where I mentioned monochromatic bright. This is a kind of sex where you have both sex, or kind of species where you have both sexes having this bright and colorful plumage. So this Chiloe widgeon, underneath the words monochromatic bright, both sexes have this green in the head, versus a Philippine duck, the one immediately to its right, under the words monochromatic dull, is again kind of a species where both sexes have this more cryptic plumage that lends itself to camouflage. And on the very bottom left there, we see the hybrid of these two species. And in this hybrid, only the male gains its green head back, which suggests we might actually have two independent mechanisms for what's controlling bright plumage. We have one in females, which we know to be estrogen, and then we have a mystery one that's in males, but it's very easily restored with just kind of one cross back into our hybrid. So we have two hypotheses here that we're testing about the evolution of this dichromatic character. First, we could have multiple gains. So this might lend itself to the sexual selection hypothesis, where you have this ducks kind of starting out in this monochromatic dull, males gaining this bright head color, probably through sexual selection, and then females gaining it maybe through something that we call the transference hypothesis, which is that females sometimes gain male-like traits. Or you could have multiple losses. And this is what we look at with more of this biological evidence. So in this case, you would start out with both sexes having this bright plumage, like the Chiloe widgeon. Females would then lose it, probably through this estrogen suppression mechanism, and then males would lose it to this unknown way of how they suppress it, probably not estrogen. So you'd have, again, this dull species at the end. But before we actually go into testing these hypotheses, I want to zoom out and kind of look at the big picture questions about what this research is getting at. And it really is about the repeated evolution of sexually dimorphic characters. And so throughout this talk, you're going to see that the evolution and the gains and losses, no matter what direction, are really a mystery in many species. And we're going to be asking essentially, are those changes driven by similar environmental pressures? So are ducks in similar environments more likely to be monochromatic or dichromatic or bright and dull just because their environments are similar? Are we looking at similar changes in the genome? So do monochromatic and dichromatic species have more similar genes or genomes than those that might be dull or bright? You know, when we're looking at these kind of groups that we're binning these organisms into, are the evolutionary processes that we see that lead them to the phenotype that they are the same or different? And I'll get into a little bit more as to what these means later. But essentially, we're looking at through time, are the changes we see predictable and similar? And the first question we're going to ask is basically how this trait has evolved through time. Before we can ask what environmental pressures or genes have led to the evolution of this trait, we have to understand how the trait has evolved through time. And this is a tree of dabbling ducks. So these ducks are the ones that kind of stick their butt in the air. They're probably what you think of when you first think of a duck. And this is a tree that's made with DDRAD data from Flor Hernandez and colleagues that is currently in submission. And really, this is just going to orient yourself to three of the biggest groups of dabbling ducks. We have the genus Spatula, who have spatula or shovel-like bills. These are our shovelers. Then you have the genus Anis. These are our mallard-like ducks. These are probably the ones you're most likely to see in your local park. Then you have the genus Mareca. These are your widgeons. These are what I call probably the cutest ducks in this group. It's actually how I teach people how to bird. I'm like, if you think it's cute, it's probably a widgeon. And then you have other basically, because ducks, again, hybridize a lot, we have sometimes a hard time figuring out their placement. And so right now, we don't have a great name for this group, but it's mostly our steamer ducks. But we're not going to be talking too much about this group today. It's just these three: Spatula, Anis, and Mareca. And to understand how a trait has evolved through time, we do something called ancestral character state reconstruction. And I know that there's a lot on this slide, so we're going to walk through it together. And I want you to start on the very right side, where you have these kind of like blocks stacked on top of each other. Those blocks represent basically a character state of a species in this phylogenetic tree. And if you've never seen a phylogenetic tree before, think about it just as kind of like a family tree in terms of blocks that are similar and branches that are similar or closer to each other or more similar to each other. And in this case, each of those blocks represents the plumage status of a species. So a red block would show that that species is monochromatic bright. An orange block shows that that species is dichromatic, and a yellow block shows that species is monochromatic dull. And as we go from the right to the left, you'll see these little circles. These circles are pie charts of probabilities that a common ancestor, so kind of like a common just relation between all of these species, had this trait. So you see that at kind of the very end of the tree towards the blocks. Most of those circles are entirely orange or yellow or red. And those tell to us that, okay, these common ancestors were either monochromatic bright or dichromatic or monochromatic dull. And we're interested in seeing kind of as far back as we can go what the status of this common ancestor was, because that will tell us the direction that this trait has evolved in. And we can see in the box there where I've blown it up, this common ancestor is mostly orange in the pie chart, which shows that it's probably dichromatic. So in this group of dabbling ducks, we have this intermediate state of dichromatism as this common ancestor, which isn't super informative on its own in terms of answering this hypothesis, but we can also see as we move through the tree that this trait just changes a lot. So each time you see that red circle on the tree, that is where you have a switch from dichromatic to monochromatic. And then each of those arrows at the end are places where you have switches from monochromatic to dichromatic. And I know I'm saying a lot of words, talking about a lot of transitions. And the biggest part to kind of take away from this is that this is just a trait that changes a lot. There isn't a predictable pattern that goes through when we just look at the tree on its own. And to answering this question about how it's changed through time, whenever we kind of have doubt or uncertainty, we can always try and add more data. So instead of just doing dabbling ducks, the next step we did was to look at all ducks. So all of Anseriformes, not just this one group. You can see the dabbling ducks kind of on their own little group towards the bottom of that larger tree. And this tree was made using Yates et al. Data. We don't have a great tree of all Anseriformes, but I'm going to talk about that a little bit later. So in this character reconstruction, moving into the good bits, we can see that this pie chart at the very, very beginning of the tree, the common ancestor all ducks, is mostly red, which to us indicates that the common ancestor for ducks was probably monochromatic bright. And this supports this biological hypothesis that you first had two, say, ducks that had like bright green heads, females that then lost this through their estrogen suppression mechanism, and then males have repeatedly lost this trait through some unknown way. But again, the main kind of takeaway is male bright plumage, this like green head and this very like fancy coloration, has been lost and then regained multiple times throughout this whole tree. So it's a trait that's very flexible and potentially could have some really interesting evolutionary consequences when we take a deeper look at that. But before we kind of look again into the genomics of this, we're going to ask if all of these changes that we see have some sort of common pattern. And these are what we call evolutionary correlates. And in this, we're kind of testing the idea of that phenotype is related to behavior. So the way you look is very much related to the way that you act. And in this case, we're testing this hypothesis about the ecology of investment. The ecology of investment essentially says that the sex that invests more in making gametes, eggs or sperm, or raising offspring is going to be the one that's going to be potentially a little more conservative in the ways that it survives. So in this example, females are the ones that are going to be laying eggs, and they're typically going to be the ones that are caring for eggs and raising for their young for the most part. So this female ruby-throated hummingbird, you see in her brown state really blends well into the tree. She doesn't totally stick out, versus the male that was sitting on the nest, that could attract predators or make it more difficult to actually raise those offspring. So in order to do this, we need a tree, which we already have. And we also need a character data set. So something that talks about these behavioral characters. And I will go ahead and use this as a kind of idea to say this is not a novel idea. A lot of people have kind of seen that you'll have a research idea when you're making your proposal and then find a paper from some guy in the 1980s that have done the exact same thing. In this case, one of the guys set in the 1980s that has done this are Scott and Clutton-Brock, who very conveniently also had a data set that I could work with to test these hypotheses, where we now have a much more robust framework and methods to get at kind of the meat of these characters in an evolutionary framework. So what we did is we did something called a phylogenetic ANOVA. And a phylogenetic ANOVA essentially looks at the correlation between multiple traits in the context of a bigger phylogenetic tree. So it leaves room for relationships as a cause of similarities. And so we looked at male care, migration, territoriality, and latitude as different traits. And we found that male care, migration, and latitude were very important. So this tree on the right is the same tree that you saw before. It's just become kind of like put on its side and sent into a bend. And what I want to show you here is on this tree where it says C, kind of at like 11 o'clock. This is a tree. Sorry, if this is a clock. Or about 1 o'clock where it says diving, you'll see that that inner ring is orange. Those are dichromatic species. That second ring is the male parental care status, and that middle shade of green means that they provide no care. And then finally, that most outer ring is our migratory status, and it shows that they are a regular migrant. And this is the pattern that we kind of see throughout the whole tree. In that dichromatic species, so species in which the males and females look different, are more likely to migrate, be in northern latitudes, and have males that don't care for their young. So essentially, the males are brighter, they would probably attract a lot more predators in caring for their young. So it makes sense that they would kind of want to get out of there for one reason or another and leave the parental care to the females on their own. On the flip side, monochromatic species, species in which males and females look the same, are more likely to provide joint parental care. So the males are more likely to stick around longer. They're more likely to help in either caring for the nest or defending the female while she's on the nest. So it really does show that this outward appearance does lend itself to different behaviors too. And that leads our then third objective left, which is really diving to the genomics of identifying the genes responsible for controlling the dichromatic plumage and understanding the evolutionary history of these genes in themselves. So for this part of our data, and part of the reason why it's really cool to be able to do this in kind of the age that we're doing, is that whole genome sequencing has become so much more accessible. And in this part of the study, we are asking if the genes controlling the bright male plumage are the same or different in closely related groups. So going back to that original group of just the dabbling ducks, not all ducks, we have each of those three main genera represented. And what we did is we took 10 individuals from a monochromatic and a dichromatic species from each of these groups. And so this is where we have the Chiloe widgeon and America widgeon from America. In Annis, we have our yellow-billed pintail and northern pintail. And then in our Spatula genus, we have Cape shoveler and northern shoveler, representing, again, our monochromatic and dichromatic species. And this is specifically where the funding from AOS went in. So I'll go ahead and say a huge thank you. Would not have been possible without that funding. And you might be asking, like, why do we need to use whole genome sequences? There are other ways that we can look at data. And the best answer to that is really that whole genome data looks at the entire genome. To the left there, you can see what is essentially a pathway of how melanin is deposited in feathers. This is in chickens, but chickens are actually pretty closely related to ducks. And you can see those like 3 letters that you might see next to the arrows in between this flow chart. Those represent the different genes that at some point are being either turned on or regulated at any of these steps in this kind of synthesis pathway. And essentially just saying that there's a lot of genes that can contribute to this. Even though it's a pretty simple trait, there's a lot that needs to work together in order for this to work. And not only that whole genome data lets us capture kind of all those genes, but it also lets us look at non-coding regions. Another reason why whole genome data is specifically good for ducks is that ducks are actually treated as a model species in any way. We have several fantastic annotated reference genomes, including for wild birds, where essentially we can be like, okay, like we know that this like essentially sequence of genes, this place is related to this gene, this other place like is related to this gene, makes it very easy for us to really pinpoint in our data what differences could be related to what genes. And third is that whole genome data can easily be shared and used for other projects. So for us to get the most utility out of this data kind of into the future, and also as for collaborative efforts, we really want to use whole genome sequences because that way anyone can use them. And as mentioned, we've already published 30 of them so far. They're on GenBank if you want to use them. So here's an example of a species where you might have differences in phenotype that are not actually due to the differences in the genes themselves, but in the coding regions between the genes. And this is also an important reason why we use whole genome data. So this was a study that looked at kind of face color in Gouldian finches. And this is a Manhattan plot. So when you think of Manhattan, you think about a skyline. You're looking for skyscrapers in these plots. And these skyscrapers indicate areas where you have differences in the genome. And you can see that you have this kind of gray box in the middle. And then up top would be something like a reference genome that kind of tells us what gene is at what location. And you can see that it's not really overlapping with any of these genes, but instead in a regulatory region of a gene. And this is really important because we already know that the estrogen suppression mechanism that females use to block their plumage is actually also due to regions of change in the regulatory regions of a genome, basically kind of these areas that tell a gene to either work or not work, or work a little or work a lot, instead of the genes themselves. So using whole genome data allows us to both put at the genes themselves and the areas that might control the genes throughout the genome. So with our whole genome comparisons, we're thankfully not going into this blind. When we're looking for sites of differentiation, these little skyscrapers, which we'll call sites of high FST across the genome, we already have some preliminary data on mallards and the mallard-like clade. So our collaborators, the Loretsky lab, already did a study where they looked at the mallard, dichromatic species, and a bunch of mallard-like ducks that are very closely related here in North America, which are all monochromatic. And this is the Z chromosome. So again, kind of like a Manhattan plot, you can see these little kind of peaks of differentiation, kind of at the same spot on the Z chromosome, which is the sex chromosome. And they align very closely to a gene called TYRP1, which is one of the genes that we have in this melanin synthesis pathway. So one of the hypotheses we can test already is, okay, in each of these three genera and all of these ducks, do we see these same patterns where you have high differentiation on the Z chromosome, the sex chromosome, and this area where TYRP1 is? And we also have for TYRP1, in addition to knowing it's in the pathway, we do have experimental studies where the expression of this gene confirms its importance in the head color there for mallards. So moving on to our results, for our first comparison on the Anis group, which is very closely related to mallards, we have lots of peaks throughout, but we do have a peak on Z, which does to us indicate that there could be, again, TYRP1 as a significant player in controlling why males are bright in one species and why they're not in another. But in our other two genera, we do not see that same peak. We might see like a little one here and there, but nothing like we see on this first group for Anis. And Spatula and Mareca, we don't see, again, that like very high peak of differentiation. And though TYRP1 might play a little bit in it, we are not entirely attributing it as like an obvious slap in the face that this is a gene that is controlling for this bright plumage. And all of this to say is that these gains and losses that we see kind of throughout actually may not be due to the same genes, even in these very closely related species. And we're going to take a couple of approaches moving on how to actually elucidate what genes we're looking at. And one of those is to make gene trees. So we're going to make gene trees of essentially all the different candidate genes that might control for bright plumage color and see if their changes in their reconstruction match what we see in a species tree. This also allows us to get at kind of the bigger question of to like, how has this trait evolved from like an evolutionary mechanics standpoint. So it might be due to something called homoplasy or convergence, which is essentially that you have similar traits evolving in similar environments, and it might be the same genes or maybe different genes that are changing. You might have introgression. Again, ducks hybridize a lot. So if you have similar genes across the genome that are causing these changes, it might just because ducks are kind of inserting their genomes all over the place, which makes my life a little more difficult, but it's a very common way that traits are kind of changed and exchanged through species. And then you have shared ancestral variation, which is essentially that the gene trees make sense. Like the gene trees are not all over the place. They're in very organized groups, but our species tree doesn't match up with these gene trees. And this is something that we call ILS, or incomplete lineage sorting, essentially showing that how we understand the history of these genes evolving and how we understand the history of these species evolving is different from one another. So this leads us to kind of the next step in what we're doing. So all of these results might change a little differently. And that's because instead of using the Yetzal tree in the future, we are building a whole genome phylogeny of all waterfowl. That photo of me is me taking a topad from a swan in our collection, a black swan. And currently we have 127 out of 175 species sent out for sampling. The other remaining ones are ones that I'm either actively collecting right now from our collection or that we're waiting on loans for. But we're hoping to get all of those at the sequencer in the next coming months so we can actually create a really nice whole genome phylogeny of all waterfowl that can be used in many applications. And I'll take this opportunity to show some of the undergraduate driven research where this phylogeny may come into play. This first photo is of Kai, who studied wing spurs in Anseriformes. So many duck species have kind of like little knives on their wings. And Kai essentially looked at, are we looking at the same bones or different bones? And how has this trait evolved through time also using these same methods? We have two new undergraduates that are starting with us. Avi is studying bill color change in ruddy ducks. And although that's only on one species, we might be able to find some cool kind of genetic history and looking at the group of these stiff-tailed ducks as a whole. And then Adrita is looking at eye color dimorphism. So we can't, using museum specimens, look at the soft part color on its own. So we have to rely on things like eBird photos in order to really take really good measurements of these colors. And she is going to be also able to reconstruct the history to see if these changes are driven by some sort of common ecological similarity. So my main takeaways for you today about ducks are that bright plumage in males has been repeatedly gained and lost kind of throughout the duck family. There's no clear pattern right now as to why some ducks are bright and why others are dull when we just look at genetic or when we look at familial similarity on their own. Second is that changes in these monochromatic or dichromatic plumage are likely associated with changes in parental care. So ducks, species that are dichromatic, the males are less likely to stick around to care for their young. And finally, in dabbling ducks, these changes in dichromatic plumage are probably not the result of the same genes, which could suggest that we are looking at convergence, where we have similar environmental pressures that are changing the genomes in different ways, but basically end to the same phenotypic result. I'll go ahead and add my acknowledgements here. Everyone on the Drexel ornithology team, lots of great funding sources that were primarily mentioned earlier. Our collaborators at University of Miami, University of Texas, El Paso, and Wright State University, and then the many, many, many museums that have contributed to this research. And I'll go ahead and say thank you. I believe we are at the 41 mark here. And please feel free to contact me at any time if you have questions outside of the symposium. And thank you so much for your attention.
All right, Emily, thank you so much for sharing the research. Also, great going through so many concepts for the audience. Really appreciate it. It was good for me to hear it too. But Uh, as we're just waiting here again, just to remind everybody, you can and people are saying great talk and excellent presentation. You can ask your question in the Q&A or the chat or the hand raising uh, as well.
I'll go ahead and say, yeah, go Gators, Jeremy. We're hanging on there for March Madness right now. Sorry, Robert. I know that
So,
you're
oh
at
yeah, we're
Duke.
here with
Yeah.
Duke. Yeah. So so we'll see if we meet at the end. Um, I'll start reading then. So Daniel Goldberg asks, do ducks show examples of bilateral geandromorphs, or half female, half male? How does that factor into your results?
So I am not totally aware of bilateral gyandromorphs that ducks have. There are examples in chickens, and again, like chickens and ducks are very closely related to one another. Um, and it's a very similar concept there. I don't know of any examples right now of like in collections of records that we have. I have no doubt that they exist. But it would be interesting to actually, I think, look at the chicken, for example, because a lot of their ornamentation, they also have the structural coloration, could be caused by these similar genes too. Um, I don't think it's going to change any results on this broader scale of asking these questions about how these traits have changed through time, but it could provide us some very interesting context of when we see individual variation, how just simple it is for this trait to kind of be changed on or off, even within the same individual.
Yes, and Matt asks, hi Matt, by the way. Uh, Interesting talk, Emily. Quick question. How did you decide on the zero point 0.6 threshold for identifying the FST peaks or for identifying peaks in the FST plots?
So those are not so much the threshold. We are going to be looking at the top, I believe, 1% of FST plots. That was more kind of a visual guide, A, for me to make sure I'm aligning the plots correctly, and B, to just kind of show it's a fairly standard measure of where you have FST differentiation, and that 1% is going to kind of change between each plot. That was more of a visual guide of saying like, ooh, like here's might where a peak is for each of these. So it's not our official cutoff for identifying peaks. Very astute, Matt.
Work in progress with the threshold. And Paul Gray has I think clarifying question. Can you please review the reasons why you think the original duck or ancestral duck was bright and monochromatic?
So I will start by saying that in evolutionary biology, we tend to go by this philosophy that the simplest answer is going to be the best. There's debate in that, but generally that's the way we like to think. And in this case, we'd think that the common ancestor to ducks probably had the simpler answer that both sexes look the same instead of the more complex answer that both sexes look different. Um, and in our case, because we know, at least in some of these birds, that the females can suppress this bright plumage, it makes sense that both sexes probably had this to begin with. As in, it would be kind of weird for dichromatism to evolve just because a female was able to have a trait just so she could suppress it. And in some other work that I'm doing, we're also seeing that more of these female-like traits, sorry, these male-like traits actually were present in both males and females. And instead of being kind of originated because they were sexually selected upon, it's more that these phenotypes are exaggerated through time in one sex instead of just being entirely absent. But our ancestral character state reconstruction had that pie chart at the end, which made us statistically think and support the hypothesis that this common ancestor to ducks was more likely bright and monochromatic.
Uh, okay, yeah. So Paul says thanks. Um, okay, yeah, I just had a question. In the fan phylogeny, one of the trait distinguished by different colored rectangles was either dichromatic or monochromatic. And earlier in the talk, there was dichromatic, bright monochromatic, dull monochromatic. So if you put those three on the fan, does anything happen or does that look?
It does not change. So I actually talked about this at Evolution of when you're doing these kind of analyses, it's very important if you're coding them as two states or three states or whatnot.
Yep.
If we change it from just monochromatic to dichromatic to monochromatic bright, dichromatic, monochromatic dull, the correlations don't change as much. What we just learned is that these monochromatic bright species are more likely to be in southern latitudes.
Okay. Okay, cool.
That's the only distinction.
Um, and I just wanted to ask, I think this is sort of your future directions, but those now I'm forgetting which ducks it was, the shovelers and the yeah, shovelers is one of them with the Manhattan plot peaks. They didn't have peaks on the Z chromosome necessarily, but there were some peaks.
Yes.
So have you looked into it yet, or that's the next step, what
So
those could be?
I've looked into it a little bit.
Mhm.
I want to take a little more conservative approach where instead of just kind of testing what we think is the more likely like way and then finding an answer and stopping there, we're going to take a much more broad approach. But in looking at the data, the one that sticks out the most is this gene called TYR. So I talked about TYRP1. There's another gene on, I think, chromosome 1 that's called TYR, where you have peaks, at least in Mareca, so these widgeons where it's kind of right there. But another thing we're going to be doing that I failed to mention is something called the phylo-GWAS. So we're going to do this whole genome phylogeny. We're going to look at the monochromatic and dichromatic species within these groups, and we're going to compare their genomes to one another in this context. That should also hopefully allow us to essentially find these peaks of differentiation and hopefully, again, get out, like erase some of this background noise that we're seeing. TYR, though, I do think is a very likely candidate. It's just kind of a gene that's earlier on in this melanin synthesis process.
So you're going to take like a comprehensive kind of approach to
Yeah, as much as time will allow me.
cool, cool. You're right, right. And then I just wanted to also ask, I hope I'm not missing anything here from other people, but so like a chicken when it gets a female chicken when it gets old, a chicken bird like galliforms can would get some male plumage characters, right?
Yes.
So uh any idea if what the ancestral state then for Gallo and Sari
So
is or
that
yeah.
that is what my undergraduate thesis was, which if any of the Florida team is on here, I promise I'm getting that paper together. So in tarsal spurs, which are these kind of like knife-like traits at the end, which females, when again they stop producing eggs and take them these rooster-like qualities, their spurs will like super grow,
Hmm.
was present in both sexes as the common ancestor too. And so again, I haven't tested plumage in this context. Rebecca Kimball's lab published a paper that I'm on that talks a little bit about this in like the fleshy trait structure
Hmm.
on galliforms. But in terms of these secondary sexual characteristics, at least like tarsal spurs, we do find that the common ancestor in this group was kind of the similar in that both sexes were the same and probably used this at least animal weaponry the same way. And then through time, females suppressed it with estrogen, and males just kind of grew this like much more exaggerated trait, probably through sexual selection.
So it may not just be plumage following that
And it may not
pattern
just be
potentially.
plumage that's
Very
following
cool.
this.
Very
Yeah.
cool. And I'll just end it with a kind of a fun thing. So so you're getting like pink-headed duck and Labrador duck for this?
We are. I believe we have pink-headed duck. We have one that we just sequenced, which we're really excited to do kind of a biogeography paper on.
Mhm.
Is I'm drawing the common name now, but it's into cherries. It's this duck that's from Japan that's also considered to be kind of like functionally extinct.
Mhm.
Um, but we have, I think, 4 or 5 species that we're sequencing that are considered either very critically endangered or extinct as part of this phylogeny.
Very, very cool. All right. So I'm looking through all the stuff. I think I got to everybody. Chris Ann has a reminder post here about the post-seminar evaluation form. But otherwise, I just want to thank everybody so much for being here. Thank Emily for her great talk. I really enjoyed it. I did not expect the ancestor to be most likely in that in the reconstruction be monochromatic. So I uh, certainly learned something here, and bright and monochromatic, I should say. And so yeah, just thank everybody for being here, and we'll be back next week.
Thank you all. Thank you, Robert, and for everyone who's organized this.
All right, everybody. See you, Emily. Bye bye.